Good evening, all. Good evening, all. You are welcome to Tutorials with From Eternity the Schoolboy. Today we'll take a look at biology. In our last class, we concluded with cell structure, components, and their functions. Where we describe how the structure of the animal plant cells and the components, the organelles, both the cell inclusions and organelles, and what they do. And we ended with the topic with the differences between plant cell and animal cells, with reference to the organelles that we find in both of them, and organelles that we find in plant cells that are not in animal cells, and the one in animal cells, not in plant cell. And at that point, we realize that plant cells, there are more organelles in plant cells that you cannot find in animal cells than the organelles in animal cells you cannot find in plant cells. These were what we discussed in our last class. So, but today we enter another topic. We go into the cell and its environment. Cells and its environment. The environment where the cell finds itself is very, very important. Is pivotal to the survival of the cell. Am I communicating? Is pivotal to the survival of the cells, because there is exchange of materials between the cells and its environment. There is exchange of materials between the cells and its environment. Even in cells as cells as a single cell, as an organism, as you see in amoeba, euglena, paramecium, as the case may be, they need exchange of materials with their environment for them to survive. True of us, they need exchange. Without that, they will not be able to do survive. That amoeba, as a single cell organism, carry out most of the activities of a full organism at system level, is because there's an exchange of material between the amoeba and its environment. If not, the amoeba will not do survive. Then, coming to organisms that are multicellular, be it system level, be it organ level, be it tissue level, as the case may be, the cells that cannot exist on their own come together to form tissues, organs and systems. Even within this system or organ or the organism, within the organisms, there is interaction between the cells and its surrounding. Am I communicating? Using human being as an example, you realize, or higher animals, you find out that the cells is bathed, the cell is surrounded by fluids. You have what is called the intracellular compartments and extracellular compartments. And the waste product of metabolism leaves the cells and go to that extracellular compartment. Nutrients, be it inorganic compounds like oxygen, like water, oxygen, CO2, there's an exchange between the cell and the environment. Without that, the cell will not do what? Survive. This serves as an introduction to what we mean by cells and its what? Environment. Am I communicating? Am I communicating? Am I communicating? Why are we so quiet? You know that our review time is always hot. Review has gone. We are in lecture now. So let's remove the composure for review. Review is a time you review what you have been doing. It's not a time to be gallivanting. It's time for you to beat your chest. I'm not doing well. I need to do what? Start what? Doing well. Or time to tell yourself, I'm trying, but I need to do what? Do more. That's what the real review is. It's not a time to be happy. It's a time to truly see that you need to do what? More than what you are doing. So now that one has passed. I want us to come together with the mind that we are normal lecture now. So if you keep taking up review, you lose the information you are going to get now. You'll be distracted, and if you're distracted, you'll be given another review again. You go back again and mess up again. You come again, I'll give it to you back again. You become angry again. You become somehow again. Doing lecture again, you'll be absent-minded again. Another review again. You go again and do nonsense again. It will continue. But if you keep review at the side and focus on the lecture, you grab this information. If you watch, most of the top questions I give you are based on what you have done. Only few are what you will still do. So listen now and put your mind together to grab the information I want to pass across to you now. So please, I want you to do that. Now, this is an overview of what the cell and its environment looks like. Even be it a single cell organism or be it a multicellular organism. Now let's go. Of course, before we do that, I want to look at what we call what is a matter? That one is no longer a new term to us, right? A matter is what? Anything that does what? Has mass and does what? Occupies what? Space. Remember those that were there when we started this tutorial for the year, for 2023/2025 session. Somehow around what time? You are there when we started for this session. That was, call it 2 months ago, right? There. We talked about chemistry, what is matter, and we end up saying that matter is what? Everything. True of us. Matter is what? Everything, both living and non-living what? Component. True of us. Matter is everything, and that's why, because chemistry, if you define chemistry, if matter is everything, chemistry is defined as the study of what? Matter, properties of matter, component of matter, blah blah blah, and the changes matter does what? Undergo. So if chemistry studies matter, that means chemistry is everything. True of us. True of us. Study of chemistry is study of what? Everything. Because everything is made up of what? At least atom. True of us. No matter what it is. Now, that's why both living, we are still in line with that. That both living and non-living things are composed of what? Matter. Now, matter is, we have component of matter and states of matter are not the same. I hope you know. Component of matter, a matter may be composed of what? Atoms, ions, or what? Molecules. Are we communicating? Am I communicating? Atoms, ions, or what? Molecules. These are the component of what? Atom. So, when you say atom and ion, of course you know the relationship between atom and ion, right? When there is what? Changing of what? Electron. True of us. Electron number, right? An ion will be formed. whether if electron is added, then a negative ion is formed, right? If electron is lost, a positive ion is what? Formed. Because atom on its own is naturally what? Neutral. It's electrically what? Neutral. So ions is formed from atom. Even molecules also form from where? Atom. Because atom combine to form what? Molecules. And there's change in electron of the atom to form what? Ions. I'm just having a summary. Now, the state of matter, we have the solid what? Liquid and what? Gas. Am I communicating? Solid what? Liquid or what? Gas. We are not going deep into chemistry. This is biology. I just want to mention this to throw important light, to throw more light on what we are looking at. Now, if matter is composed of everything and the state of matter is solid, liquid, gas. Remember when we talk about the kinetic theory of matter, in a way we talked about what happens in solid, what happens in liquid, and what happens in what? Gas. Am I communicating? That's the particles that form liquid, solid, that are held together by what? Forces, right? That made it to be fixed, to be in a fixed what? Position. They only do what? Move about a fixed what? Position. If they must vibrate, but they are still there. But in liquid, when temperature increases, you know what happens now? The particle will get more energy and they break the what? Intermolecular forces and become a bit what? Loose, right? While in gas, more increase in the temperature, heat, vacuum temperature will lead to what? The particles that will now separate more intermolecular forces will what? Broken. The particles will do what? Separate what? More. This thing is also important in the movements of materials from outside a cell to inside a cell, and from inside a cell to what? Outside a cell. That's why I want to bring our mind to that. Because the two major processes through which substances, through which materials are exchanged between the cells and its environment, the two major ones are what? Osmosis and what? Diffusion, right? Osmosis and what? Diffusion. Good. But generally we have four processes. We have osmosis, diffusion, active transport, and what? And what? Passive transport. Now, we'll go to that level, but I want to explain also some other things. Now, having known what and what that can process through which these materials enter and leave the cell, I want to say something on the type of environment the cells can find what? Itself. The types of environment the cells can do what? Find what? Itself. Now, sometimes the cell can find itself in environment that, of course, fluid environments. Let's use cells in higher animals as an example. A cell can find itself in a fluid environment, and that fluid is highly what? Concentrated. Am I completing? Even more concentrated than the what? Inside of the what? Cells. That it has more solutes, right? Than the inside of the what? Cell. Now, if a cell finds itself in such an environment, the cell is said to be in an what? Hyper what? Tonic what? Environment. Okay? Hyper what? Tonic. Hyper means high. Tonic, when you put a solute, that's what we call tonicity. That's what we call what? Tonicity. When you put a solute inside water, am I communicating? Osmotic pressure is exerted by that solute, depending on the concentration. Now, it will add that increase in osmotic pressure is expressed as increase in what? Tonicity of the what solution, because you know that previously we have solvent. When you add the word solute, it becomes what? Solution. Now, if the concentration is high there, or what, nothing, you don't even need for it to be high. Once a solute entered, the thing is no longer a solvent, because the solute now will add what is called tonicity to the what solution. Now, but the higher, the more solute you add in that small solvent, the more tonicity, the more osmotic pressure, and the more tonic the liquid becomes. So if the tonicity in the liquid, in the fluid outside the cell is higher than the tonicity of inside the cell, that in that situation you say that the cell is in hyper tonic environment, or the is outside, the fluid outside is hyper tonic. Now, by the time we enter osmosis, I'll explain what will happen when it's in that hypertonic. I'm just giving you the type of environment the cells can do find itself. Now, if the cell find itself in an environment that has less concentrated in terms of solute than the concentration inside the cell, that means the one outside has less tonicity, is less tonic, because the cell itself alone, by having, you know that the cytoplasm is fluid in nature, right? Remember we discussed, right? And the other dissolved and suspended particles inside the cells, these things add tonicity inside the cell. But if the cell, the one outside the cell, is less tonic, by now you understand what I mean by tonicity, right? That's why I explain it here. If the one outside the cell is less tonic than the inside the cell, you say that the cell is in an hypo, meaning lower than, in an hypotonic environment, or that the fluid around the cell is hypotonic. Now, but when the cell finds itself in an environment in which the tonicity of the fluid surrounding it is the same thing with that inside the cell, you say that the cell is in an equal tonic fluid. That's the meaning of isotonic. So iso means what? Equal. Hyper means what? Higher. Okay? Or high. Hypo means what? Lower than or low, as case may be. This is the three types of environment the cell, with respect to the concentration and tonicity, the cell can find itself. These things have their implications. These things have what? Implications. This thing has what? Implication. On a normal, it's better for a cell to be in a what? Isotonic what? Solution. But when we go into osmosis, I will point out the impact of these things to the cells. Okay? Even though hypertonic is not that good, but there are situations anyway. That one is applied when you come up. For those in pharmacy mainly, this thing has applications, because sometimes you can actually intentionally give a drug. Okay, let me not go into drug. We are in biology class, but just know that by the time we enter this osmosis, at least I'll use the opportunity to say one or two things. All right, so this is what the type of, remember the topic, cell and its what? Environment. That's why I went to the environment the cell can do what? Find itself. This is the type of environment the cells can find itself. Now, let us now go into these processes through which the cells exchange materials with its what? Environment. Let's take them one after the other. one after the other. Okay. There are four major methods through which materials enter or leave the cell. Four major ones. We have diffusion, osmosis, active transport, endocytosis, and exocytosis. Diffusion, osmosis, active transport, endocytosis, exocytosis. In fact, in some materials you will see they classify it into active and what? Passive. We have passive, diffusion and osmosis being under what? Passive, because they don't require what? Energy. While active transport require what? Energy. If you go up a bit advanced level, you start seeing facilitated diffusion. That one is beyond your distance. This is just O-level class. You start seeing things like facilitated diffusion and other things involved. Let's limit ourselves to what? Diffusion, osmosis, endocytosis, cytosis. So let's take it step by step. Maybe I'll still mention some other things you need to know, but just know that these are the basic one you need to know based on your syllabus. Okay. All right. Now let's go to diffusion. We were talking about diffusion since. We talk about diffusion in chemistry. Talk about diffusion in physics. We're talking about diffusion in what? Biology. If you watch now, diffusion in biology now is going more biological. Is going more what? Biological. While diffusion in physics is going more what? Physical. And diffusion in chemistry is going more what? Chemical. But all of them are what? Diffusion. Now here you mention more of the body parts or part of organism with respect to what happened in the what? Part of what? Organism. But we list some examples in what we did in physics. Now, what is diffusion? We defined diffusion before now. And diffusion is what? Who can define diffusion for us? What is diffusion? Not who. Comes what is diffusion? Fast. What is diffusion? Come see. We are listening. Water molecule. Nekwa na anya o. From region of where? Higher concentration of what? Higher concentration of what? That's the basic understanding of diffusion. I want you to know. Higher concentration of. She said that diffusion is the movement of molecule from region of higher concentration to region of what? Lower concentration. My question is now, higher concentration of what? Of the ions. Which ions? Who can answer the question? Higher concentration of what? Maybe someone know higher concentration of gas or liquid. Who can answer the question? Higher concentration of what? Michael, Chinwe, Kamsi saying that the diffusion is the movement of molecule from region of higher concentration to region of lower concentration. Ozayan, Oto. But I want to know higher concentration of what? Higher concentration. Yes. Of course, I agree with you. In concentration gradient of which higher concentration of what? There's a point I want you guys to make. Yeah, you can easily memorize a definition without even knowing what it means. Higher concentration of what? It's a simple answer.
Molecules.
Which molecule? Water molecule. Which molecule? Solute. Which solute? You see? No, it's okay. This is not review. This is what? Class. In class, you say it, I will explain. But I like it. Point it out. Because after today, you will know what diffusion is. Knowing it is different from memorizing what diffusion is. Are you aware? You know what it is. I don't want to go into thermal process. Yeah, long story. But I want to just basic understanding of what diffusion is. Now, diffusion is the movement of what? Molecule of a substance, right? From the region of higher concentration to the region of what? Lower concentration. Higher concentration of what? That substance. Where that substance is more concentrated, right? To where the substance is what? Few or not even at all. True of us? True of us? You are here. You are here. Inside your tummy is filled with egg and a lot of things you eat. Egg, milk, beans. You ate egg, you ate beans, you drank milk, everything. Your tummy is doing your lecture is going on. You sit here and do your like this and release the bomb. Here where you are, the bomb you release is more here, right? Right? With you the release, you do like this. Give it few seconds. The bomb was not there before, right? The bomb will leave here. This is the region of higher concentration of your bomb. Two of us to the region of lower concentration of your what? Bomb. So it will move from the region of what? Higher concentration of that substance in question to the region of what? Lower concentration. And it will keep. Do you know why it moves like that? Do you know why it moves like that? Chinwe said it now. She said concentration gradients. There's a gradient, slope, gradient from the higher level to a lower level. There is concentration. So as it's moving from here going down, the order of your bomb at that side will be coming on two of us, to coming on two of us, to coming on two of us. At a point, the order there and the order where you release it are what? The same what? Level. We call it what? Equilibrium, right? They become what? Equal. No net movement from one region to another region. They'll just be what? Circulating around that place. That is what diffusion is what? All about. It can occur in gas. It can occur in what? Liquid. It can occur in what? Solid. But diffusion in gas is faster than that in what? Liquid. Do you know why? Do you know why? Do you know why? Because remember how I started this topic, introduction. I talked about state of matter. I reminded us of the one that is where the particles are what? Are what? The particles are what? The molecules are what? Separated because of the weak intermolecular forces are being broken, right? Do you know why? Do you know why the diffusion is faster in gas medium, example air? Do you know why it's faster? It's faster because the molecules are spaced out. Two of us, they spread, they move. So as they move, whatever you release from your bomb is spreading. Two of us. But that of water molecules, they are not too fixed as a solid, but at least they move around. Two of us. 1 is slower than that of what? That of what? Gas. But that of solid is the slowest. But at the end, there is movement from region of higher concentration to region of what? Lower concentration until equilibrium is what? Does it make sense? That is what diffusion is all about. Anything you are referring in the diffusion, you are explaining just built upon what I just described now. Any other thing you want to talk about in diffusion is just built upon what I just said now. So that gradient, you can call it diffusion gradient or concentration what? Gradients. Diffusion what? Gradient or concentration what? Gradients. Okay? I mentioned thermal. In fact, in physics was defined. In physics, diffusion is defined as a thermal process. True of us. In your physics, when we define diffusion, it's defined as a thermal what? Process. Can you remember? Okay, we have not done the diffusion here, right? Have we? Okay. See how your physics define diffusion. I want to just say one or two things there. I'm not going to physics, but I want to leave something there. See how physics define diffusion. Okay, physics define diffusion as diffusion is the tendency of molecules to migrate and fill an empty space due to their random thermal motion. Due to their what? Random what? Thermal motion. Do you know that heat is required to cause a change in state from solid to what? Liquid to what? Gas. True of us. And we say that diffusion is higher in gaseous state than in liquid and solid what state. So that means this implies that the higher temperature, the higher the temperature, the higher the what? Diffusion rate. True of us. True of us. The higher the temperature, the higher the what? Diffusion rate. So temperature actually increasing temperature actually increases what? Diffusion rate. That means diffusion is a thermal what? Process. It's a thermal what? Process. Thermal heat. It's a thermal what? Process. It's a thermal process because it's affected by what? Temperature. Diffusion is affected by what? Temperature, not the what? Concentration. Okay? Diffusion is affected by temperature, not the what? Concentration. So if you change the concentration of a particular substance here, it will not affect the what? The what? Diffusion. What affects it is what? Change in what? Temperature. Hope you know that concentration and concentration gradient are not the same. I hope you know. Are you aware that concentration and concentration gradient are not what? The same thing. Concentration gradient is a kind of, it's relative, it's a ratio, true of us, of concentration here and that. The rest, I hope you know. So it's not the same thing with what? Concentration. If okay, but the higher concentration gradient affect diffusion, true of us. Concentration gradient affects what? Diffusion. If the concentration gradient here is higher than this one, the tendency for the diffusion to occur is the true of us. If there is no concentration gradient, how will it go? How will it go? There will be no net diffusion. They just be roaming around, true of us. But the one that is a gradient, once gradient is there, bam, boom, and also increase in temperature. But basically, what affects it more is what? Increase in what? Temperature. That's why diffusion is a thermal process. But concentration on its own, no, does not affect what? Diffusion. Note these points. Now let's continue. So there are a lot of things that one or two things you can do. I think I've given you an example of the one that we do every day. Three of us, almost every day. I don't think there's anybody here that I've not done that today. We do it every day, right? Nobody can deny that fact. We do it every day. So that is a typical example of what happens in what? Diffusion. Am I communicating? That is a typical example of how diffusion takes place. Now let us not waste time talking too much on this. Now, are you aware? Before I ask this question, who can define osmosis for us? Who can define osmosis? Who can define osmosis for us? Who can define osmosis for us? I'm listening. Before you define that, does diffusion require medium? Material membrane for passage. No membrane required, right? Okay, good. Now, what is osmosis? What is osmosis? Movement of what?
Molecules or ions from region of lower concentration to a region of higher concentration through a semi-permeable membrane or tape weaving or something.
When I was in SS1, when was that? 2019. That was 19 SS1 first term. The picture is clear, it's still clear. One Mrs. Atuchukwu. That woman opened my eyes in biology. That was Atuchukwu. Do you know her? You know, as a biology teacher.
She was a teacher for teacher principal. She's now school
Do you know what subject she's teaching?
She did a lot of subjects.
Okay. I think she used to wear Agra at that time. Very, very sweet woman being, disciplined woman being.
Yes.
She's dark, not fair.
She's not too bad.
But not fair as in fair. If we are a very disciplined woman being, what she taught us in biology today is still clear. I'm talking about first term biology first was 1997. I wrote my junior WAEC in 1997. So SS1 by September 1997 we entered SS1. That woman, I don't know where I will see her again. Where is she now?
She's in my school district.
Where? 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 Amici. I seriously, I would love to see that woman again. You know what somebody taught you in 1997? He's how many years old? How many years now? 1997. How many years now? 27 years, right? 26 years. Still clear, crystal clear. It's not vague, as if she still teaches. She's just here now. Most of the things you see me teach you here, discipline, I learned it from her. You dare not do nonsense in her class. No. You dare not do that. No. So she told us that we are defining osmosis, that this thing you define her. She said no. If you define that in our exam or test, 0. She will ask you first. Let me Let me speak like her. Oh yeah, define that thing again. Just define it.
Osmosis is the movement of molecules and ions from the region of lower concentration to the region of higher concentration through a solution membrane.
She will ask you, what from region of what concentration are you talking about? Because osmosis can be defined in two ways. True of us, you can define it from the angle of the word solute. You can define it from the angle of the word solvent.
Yes.
So, what are you talking about now? Solute. Good. Osiki, you need to mention the one you are talking about. You cannot just say osmosis is the movement from lower. No. Lower concentration of what? Higher concentration of what? You must mention it in her exam or if once you enter class. Before you enter her class, you must study now. As you enter, she will point to you. She start from question what she has done, she did in last. Before you define it, if you define it, you must put all these things. What you define is correct too, but I want to point out something. Okay, don't worry. By the time I describe what I want to describe on the board now, you understand where I'm going to. Okay. According to what she said that osmosis is what? The movement of what? Sussex child is well. But if you're feeling sleepy, please walk around, okay? What is define it again, please? Let's hear you again once more. Okay. Before I go into osmosis, are you aware that osmosis is a special type of diffusion? Are you aware? It's a special type of what? Diffusion. Osmosis is a special type of what? Diffusion. But what actually happens is that doing diffusion, doing osmosis is only the solvent that moves in osmosis. Is only the solvent that moves. The solute doesn't what? Move. Anyway, in a classical definition, it depends on the nature of the membranes. Anyway, I will explain what I mean by that. The nature of the membrane also contributes to if the solute will do what? Move. Now, remember we have permeable membrane, right? I want to classify types of membranes. We have permeable what? Membrane. We have semi-permeable what? Membrane. We have selectively permeable what? Membrane. Please, I want you to know the differences between these membranes. I will still go back to osmosis, but I want to talk about this because the nature of the membrane now will guide you to understand this better. We have what is called permeable membrane. In permeable membrane, everything moves, everything goes, everything enters. Am I completing? In permeable, both the solute, both the solvent, everything moves to and fro. This going, enter, come out. That's a permeable what? Membrane. Can someone give me an example of a permeable membrane? Does God give me an example in living organisms? Eh? You don't give me an example of a permeable membrane. Example of a permeable membrane. Cell wall. Cell wall. Cell wall is permeable. It's not selective. It's not semi. It's permeable. Now, after permeable membrane, we have selectively permeable membrane. We have selectively what? Permeable membrane. Selective permeable membrane allows, it select what passes through, okay? In that one, just select mainly water and some ions, okay? Water and what? Some ions pass through. In a selectively what? Permeable what? Membrane. But in a semi-permeable membrane, semi-permeable membrane allows only water to pass through. Water only. So selectively, just select. You can allow some molecules to just enter, select. But semi is only permeable. Anything enters. So know this point. Now let's go back to what? Diffusion. Definition. In order for you to understand this, there's something I want to do. There's something I want to do to make you understand this better, not just to memorize it. I'm coming. Now, let's assume this is a membrane. Okay? This is either selectively permeable or what? Semi-permeable. Am I communicating? Now, here is, let's assume this is inside here is cell, okay? And here is the what? Environment. The cell find itself. By the time we come up, those that have already entered school, by the time you start doing your biology, physiology, you appreciate what we are doing now. When you to help you to understand your what is it called? Homeostasis and analysis. But let me not take you there. Let's continue. Now, this is the environment, right? And this is the what? The cell. Now, here you have, let's assume that this is an hypertonic what? Let's assume it's an hypertonic what? Solution. Now, this is inside here. Here is the what? Solute. Hyper. Then this is solution. Hope you know. Solute and the what? Solute and the what? Solvent form the solution outside the cell. Am I communicating? This is the solute, right? This is the what? Solvent. Two of them form what? Solution, right? And this solute impacts tonicity in this solvent. Once you introduce a solute inside a solvent, it will impact what? Tonicity. It will make it to be what? Tonic. Now, This is outside the cell. This is inside the cell. Let's assume here you have smaller what? Solute, okay? Then this larger what? Solution, okay? Solution and the what? Sorry, I mean solvent, right? Which is water, we know now. Mainly, the solvent inside the system is what? Water. Hope you know. We dissolve solute and other things. Hope you know. Both the one that are suspended or the one that are dissolved. But the major solvent there is the base liquid there is what? Water. True of us. Now here is what? Solute. Now look up. This is a semi-permeable what? Membrane. Watch out. Let's look at water. Let's look at water. Water here. Look up. Look up. Look up. Look up. Water here and water here. Which one is more? Water outside it. This is an hypertonic. This is inside the cell is what? Outside the cell is more concentrated than inside the cell. Let's assume. Now here. Water here and the one which one is more? The one inside the cell is what? More water, right? Less water, right? Look up. This is a semi-permeable membrane. This solute cannot move in a hope you know. In a semi-permeable membrane, the solute cannot move, right? The solute will remain where they are, right? Good. Now see what happens. Water is more here. Because water is more here, some of us think that it is solute here that actually attract water. That's what you think, right? Now water is more here and water is less here. So there is a concentration gradient of water. True of us. Less water, more what? More water. There is a concentration gradient. So more water is more here. Now define diffusion with respect to this water molecule. Define the diffusion. Mention the water molecule. Go.
Okay, movement of water molecules.
From where?
From higher concentration of the solvent to lower concentration.
Is that not diffusion? Is this not the diffusion? At the same time, is what? Osmosis. True of us. True. That's why they say that osmosis is a special type of what? Diffusion. Inside osmosis is what? Diffusion. See gradient now. Water molecule. See, water molecule is higher inside the cell. It's leaving the cell to where it's lower so that it will come up and normalize. In so doing, the entire concentration of the two cells will become what? Equal. Water molecule is actually moving across its own its own what? Gradients. From here, higher to what? Lower of water molecule. But the one you know since you say that osmosis is the movement of water molecule from region of lower concentration to region of higher concentration. You are defining it with respect to what? Solute. But solute doesn't move. Did it move? It's water that moved from region of its higher concentration to region of its lower what? Concentration. Define diffusion with respect to water molecule. Diffusion is the movement of water molecule from region of its higher what? Concentration to region of its lower what? Concentration. That's why we say that osmosis is a special type of what? Diffusion. Inside osmosis, the diffusion, the solute is where they are. They're not moving. It's the water that moved across its gradient so that it will water molecule will come up. If you come up now, the entire concentration, the concentration is here will now be equal to concentration here. Is it clear now? Is it clear? So based on this definition, you can define osmosis in two ways. You can say that osmosis is the movement of water according to what Atuchukwu taught us that time. I don't forget that definition. She said, see what Atuchukwu, when we were defining osmosis, Mr. Atuchukwu told us that osmosis is the exact words, I'm not missing it. Osmosis is the movement of solvent molecule from the region of its higher concentration to the region of its lower concentration through what? Selectively what? Permeable what? Membrane. That was her exact what? Words. It's in my note of 20-something years ago till now. Even though I don't know where the note, I don't know who took that note. Now, but basically, this is what happens. Now, but you can also define osmosis. But here is this. Are you the membrane I use here is semi-permeable membrane. Hope you know. Selective membrane is also that was some will say selectively or semi-permeable what membrane, but not permeable what membrane. Now, look at this one now. With respect to the solute, you can define osmosis as the movements with respect to the solute. Now, the first one I define is with respect to the what? Solvent. With respect to the solute, you can osmosis is defined as the movement of water molecule or solvent, as case may be. Solvent is better because it must not be water molecule. Hope you know. It must not. But solvent is the best to use. Hope you know. Movement of the solvent molecule from the region of lower concentration of the solute. Am I communicating? Look up. So you understand what you are writing. Look up. The first definition says movement of solvent molecule from the region of higher concentration of the solvent to the region of lower concentration of the solvent across a what? Semi-permeable or selectively permeable what? Membrane, as case may be. With respect to the solute, osmosis defined. With respect to the solute, this place is here more water or high water what? Concentration, right? You know why we are using water here? Because the solvent here is what? Water. But the best thing is to use is what? Solvent, okay? Here is what? Less what? Lower what? But in terms of with respect to solute, here is this side is higher concentration of what? Of what? Solute. Hope you know. With respect to solute, here is higher concentration of what? Solute. While here is lower concentration of what? Of solute. So with respect to solute, you can define osmosis as the movement of solvents, example, water molecule, from the region of lower concentration of the solute to the region of higher concentration of the what? Solute, through or across a what? Selectively permeable what? Membrane or semi-permeable membrane. Is it clear? That's why I ask you when you define it. I want to be sure you know what you're talking about. Higher concentration of what? You must mention it. With respect to what? From the region of higher concentration, movement of solvent molecule, example water, from the region of higher concentration of the solute to the region of lower concentration of the solute. You must mention it. Through or across a selectively or semi-permeable what? Or semi-permeable what? Membrane. So these are the two. Any of them you choose to use in your exam, you is correct. But you must mention what you are defining, the one you are using. It's very important to do that, okay? But if you just say moving from higher concentration, high concentration. That is not a good definition. Those people that were writing WAEC, it's not a good word definition. You lose marks if not scoring 0 because it makes no sense. Okay. Now, it's 6 o'clock already. So I think we have actually introduced you now. Do you now have a deeper understanding of what diffusion and osmosis are? So do you now understand why I say that osmosis is a special type of what? Diffusion. Inside osmosis is what? Diffusion. But this is for selective same membrane, hope you know. But for permeable membrane, both the solute and the solvent move. You are aware now, right? You are aware about it. Don't worry, by the time we'll do osmosis in plant, because we'll do osmosis in plant and animals, how it can apply, then you understand it better. I think it's better we'll stop here. So we can any question, ask any question if you have. Please do your what is it called? Do your review, please, so that you not come here tomorrow and be angry with yourself. Okay? Do your review. Research well. Please. It's very important. Do your review. Research well. Career for success. What is it? Any problem? Okay. Do your review, please. I beg you. I think biology is how many questions, sir? How many questions? Miss Oma. 3 questions. Okay, you not checked. Okay, now. I don't think it's up to 4 questions. Please, I want you to digest those things. Study them very well, not just look for the answer and the 3 questions. So do it very well. Go through the topic by yourself again before you can do that thing. If you want to do a good job, get your textbook, go through the topic again. Have a basic understanding of it again before you can solve it. All right, thank you very much for your attention. God bless you.